this, we're taking 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 with a pinch of salt.
Welcome, ladies and gentlemen. Welcome to another episode of A Pinch of Salt, the podcast that tries to answer the simple yet difficult questions with sensible answers. Also, the podcast where we try to present the novelties that are out there for you. And today we have a real treat because we'll be talking about Imlifidase, a new drug for kidney transplantation, a very interesting new drug. And we are very lucky that we have today with us an expert from kidney transplantation, Professor Rainer Oberbauer from the Medical University in Vienna. So you can expect to hear how Imlifidase works, how good it is for allosensitized patients, what are the tricks and tips of using this drug. So, Rainer, welcome to A Pinch of Salt.
Well, thank you very much for the invitation and for the opportunity to speak with and to your audiences.
Well, the pleasure is all ours. So, when I first heard about Imlifidase a couple of years ago, it sounded like a drug of absolutely incredible potency. So to go from normal IgG values to virtually zero in a matter of hours was until recently unheard of. So how does Imlifidase work and how long does its effect last?
Well, maybe starting the point that most of the bacteria releases some enzymes to prevent your immune system from killing the bacteria immediately. And also the Streptococcus pyogenes has an enzyme, which now is called Imlifidase, which is an enzyme that cleaves, it's a cysteine proteinase, and the way it works in the immune system, it cleaves the hinge region of the IgGs at the more distal side, specifically between two glycines. And it's very specific for IgG and does not attack the other isoforms like IgM, IgA, or IgE. The half-life of Imlifidase, the plasma half-life is quite short, and the effects are immediate. So within 30 minutes, almost all the IgGs that are circulating, and probably more, also those in the tissue and lymph nodes, are cleaved. However, the elimination half-life of Imlifidase is some more like 2 to 3 days. And Imlifidase would also kill monoclonal antibodies that are used therapeutically, and therefore there need to be at least 3 to 4 days in between, otherwise it renders the therapeutic antibody useless.
So it really works well, but only on IgG, and its effect doesn't last very long, right? The IgGs come back pretty quickly, if I understand correctly.
Yes, so usually within, I would say, 30 minutes to 1 hour, all the IgG isoforms are cleaved, so IgG 1 to 4, and then they reappear within 4, 5, 6 days, depending on the co-immunosuppression and other things. But so you can expect an immune response within, I would say, 5, 6 days at least.
So it works fantastically well, extremely powerful, but really for a short amount of time. But that is still useful for transplantation. It still helps, right?
Yeah, so the idea behind this is it gives the treating physicians the opportunity for 4, 5, 6 days where there is no alloimmune response, at least in the humoral branch of the immune system, that doesn't attack the graft. So in other words, people who are highly sensitized, meaning that their calculated panel-reactive antibody level is above 99% or 99.9%, and they never would be able to be retransplanted because there wouldn't be a matching organ available, would then have the opportunity to accept an organ where maybe the not so strong antibodies are delisted, so that there's an offer, and then you have an opportunity, a window of opportunity for about a week that you can manage the rebound of the IgGs, usually with immunoadsorption or plasmapheresis, RVIG, or maybe in the future others such as CD38 antibodies or the C20 antibodies. but it gives you the opportunity at least to receive an offer, do the transplant, and then you have 1 week to kind of make sure that the response is not taking the graft.
So it will be like changing a positive cross-match into a negative one for long enough for the immunosuppression to kick in a way.
Yes, absolutely. So the initial question was, is it wise to convert a cell-based cross-match like a CDC cross-match, or shall we go for the not-so-strong flow-positive cross-matches, or which can be converted, or is it just the DSAs, because you can do transplantation against DSA barriers also without imlifidase, and there's a long-standing history on that. However, I think it's wise to really think which of the unacceptable antigens you delist. What I do not recommend is to delist repeated mismatches against previous grafts, because there are certainly long-lived plasma cell repertoire that will come back immediately, and also probably none against known pregnancy caused antibodies, and antibodies that are very, very high and where there's a very high likelihood that they reappear. But I would recommend of speaking that or discussing that in a group together with people from the HLA lab, so that there's consensus which to delist.
So to delist is actually to take off the list the antibodies against antigens that have been present,
yes,
but not the ones that you mentioned, the most difficult ones, I guess.
Yes, because otherwise it wouldn't make sense, to kind of delist repeated mismatches, where there's, as we know, it's one of the strongest predictors of immediate, immune responses after transplantation.
A lot of memory cells.
Absolutely.
So how successful is this approach for these highly sensitized patients, these often young patients who already had 1 or 2 transplantations are desperate for a third one, for example? How successful is this approach now with imlifidase?
Yeah, I think we have to go back a step, because there are other programs for those patients in most of the OPOs or organ sharing organizations. For example, in Eurotransplant, it's called the Acceptable Mismatch Program. But if you're so sensitized that your donor frequency is way below 1%, and we speak about 0.1%, so that you have to have the chance only in 1,000 donors to eventually have one matching recipient organ, then many people that are so sensitized are waiting for a very long time, even in the AM program, and virtually never receive a graft. And so they are desperate, which I understand, and we need to look for solutions. And I think we now came up with one of the solutions. Doesn't make the sensitization go away, but as I said before, it gives you the opportunity for a week or so, at least peri-transplant, to adapt accordingly. And so what I would not, as I said before, delist is those where there's a high likelihood of immediate rebound.
So what's the most common induction therapy received by highly sensitized patients treated with imlifidase? Does imlifidase treatment change the induction therapy?
Yes, and I have to say, currently, because the previous question was about how many patients have experienced transplant and were successfully transplanted under imlifidase. Mm-hmm. And in the first studies, it was close to 50 or 53. They are now 5 years out, and they have over 80% graft and patient survival after 5 years. It was recently reported by Stan Jordan at the ATC meeting in Philadelphia in May this year. So I think it's justifiable, it's acceptable. It can also be thought about doing it in a live donor setting. It need not be in a deceased donor setting.
So the induction therapy?
Yeah, so the induction therapy is, since currently there's a study ongoing, deceased donor transplants, there's a study protocol which needs to be uniform in order to be a study protocol. And the study protocol consists of using ATG 4 days after imlifidase to prevent cleavage of ATG. And I think in total it's like 6 milligram per kg in total for thymo. Then you have the regular triple therapy with tacrolimus, CellCept, steroids. And the steroids in the first days, if I remember correctly, are in the range of like 100 to 250 milligrams per day, with a taper then thereafter. And then there's a mandatory rituximab, which I think I would not subscribe to now if I would it off of the clinical trial. And then there's also the option to do complement inhibition, either with eculizumab or the others, which I think is also probably not necessary in all cases as prophylaxis. And for treatment, I think it's a little bit different. And then there's a mandatory IVIG dosing, I think 2 times within 2 weeks in a part of a rather high dose. I think it's like 2 grams per kilogram.
So you wouldn't subscribe outside of the research setting rituximab because you think it's not necessary, or it wouldn't work, or it doesn't work? Why do you think it's...
Yeah, because I think it doesn't work. I think there's not really good evidence. The same is true for IVIG, that the antibody rebound or the plasma, long-lived plasma cells are tackled by those interventions. This will change over time, I think, but I think CD20 antibodies are probably not the way to go.
So are there any side effects, common reactions, safety issues with imlifidase? Are there any problems that one would need to be attentive about?
Yeah, really surprising to me is that it is completely well tolerated. So the patients I always see myself, there was not a single adverse event or not even nausea. So within 30 minutes the IgGs are gone and the patient feels well. So compared to ATG use, it's actually very well tolerated. One has to, or it's of note, however, that you have a lot of immunosuppression. So you probably should have, and in the clinical trials that's the case, have some prophylaxis, the usual CMV prophylaxis, but also trimetoprime sulfmethoxazole for some weeks for PCP prophylaxis. And one of my patients actually, almost after 3 months or so, developed PK nephropathy, and then it's a little bit of a challenge because usually you but reduce immunosuppression in order to cure the PK virus, and that's not so easy done in somebody who is has a strong immune drive against the allograft. So that's maybe to be taken into account.
Yep. Is imlifidase, to your knowledge, used in other research settings right now? Because as a clinical nephrologist, one would obviously immediately think about a good posture syndrome, for example, or maybe difficult ANCA vasculitis cases, or perhaps even antibody-mediated rejection. Are there any other research settings?
Yeah, you're well prepared for this talk. So in fact, there is a parallel trial on good pastures disease, but it's such a rare disease that it's very hard to come up with a sufficient number. And one of the other colleagues that you invited for your Slovenian meeting actually enrolled 2 patients last night, he told me.
Okay.
Kronbichler from Innsbruck. So it's used, and the study is ongoing now for a longer time because, as I said, good pastures is so rare that it's very hard to find sufficient number of patients, specifically if you want to do it as in the study, doing randomized and blinded against the placebo. The treatment of antibody-mediated rejection has been studied, and it will be published soon, but I can tell you it was not very effective. I mean, in terms of it cleaved the antibody, but then the reappearance, was the same as before. So probably that's not an indication. ANCA vasculitis is interesting, and we always thought about, ANCA vasculitis as one, or depending whether PR3 and the MPO are considered 2 different ones, have very defined antigens with probably very defined epitopes. And in transplantation, you introduce thousands of different new epitopes, and you can manage that with a minimal immunosuppression of a triple therapy. And so I was curious why ANCA vasculitis is still a problem, because you would assume that it's very easy to get rid of the autoantibodies against these 2 enzymes. And I think maybe such as in rejection, like for example CD38 antibodies would be good candidates, or as I said before, maybe to cleave at least, give you a window of opportunity, cleave, because you cannot be so selective. Obviously with imlifidase, it's cleave all IgG 1 to 4, and not only the auto-ally antibodies, certainly. But it's not a permanent solution, because again, after a few days or weeks, you have reappearances.
I completely understand what you're saying, and I agree with you. In my limited clinical experience, I would say that in most cases nowadays, vasculitis is not a huge problem for older patients, which is the majority. But in the ones that I did see that are very young children or very young adults, it can explode like a bomb. And in those cases, you really want to get something that works extremely fast, and that's possibly the case where one might use this, but you could never. It would be very hard
I
to
see
do a
your
study.
point, and I completely agree. If you have a severe, also pulmonary manifestation together with the lung, and if you have severe hemorrhages and you give tons of steroids and you don't see any improvement within a few days, then eventually it could be considered, I guess. But giving the avacopan trials, the Advocat trial, these are all kind of stable, as you said, patients. The median onset of ANCA vasculitis is between 65, 70, and it's very rare that you have very young people. So it's really the exception. And but on the other hand, imlifidase has been shown to be safe. Whether it's efficient also in autoimmune disease, we'll see in the good pasture setting.
Perfect. So the path has been laid. That's brilliant. So thank you. We're finished with imlifidase, and now to the tough question, which we ask all our guests, which is a getting to know you type of question. Which book, 2 songs, and luxury item would you take to a desert island?
Well, I can tell you what book I currently read. It's one of my favorite authors is the Austrian Stefan Zweig, and the way he writes, he condenses all his condensed all his manuscripts from many thousand pages to a readable 300 something. So each sentence is really, really precise. And there's a reason why he already in his very young age became very famous. But then, he actually endured these 2 world wars, and at the end of mid-60, he was completely desperate in exile, and basically conducted suicide together with his wife in the exile. But before he died, he wrote a book. It's called The World from Yesterday in English, and in German it's called Die Welt von Gestern. And it's amazing because he had contact with all the intellectual people, in the last centuries, from Freud, and they discussed, he was a pacifist. He said, it's a little bit like in the beginning of the Second World War, the appeasement maybe, and not killing so many people in the First World War, but Freud kind of convinced him that what he writes in the book, that's not the solution. Anyhow, so the book I really currently read, I'm very impressed, as you imagine and notice, is The World from Yesterday from Stefan Zweig.
That's great. Really, Vienna at the end of 19th, beginning of 20th century was really a center of thought. Many
and also
philosophers.
It was a center of arts and culture. Like he had contact with the famous painter like Gustav Klimt, and so this was really melting a crystallization point of intellectual heights.
Definitely. So what about the 2 songs?
Well, to be honest, I'm not too much into pop songs. I'm more annoyed if I listen to radio in the car or so. Sometimes, if I'm in the mood, I listen to piano music, and there, for example, the Bach Goldberg variations, the kind of, I could hear them for many times.
The gold standard.
The gold standard by the different interprets. It's interesting how different people play it differently. And the other song, if we speak about contemporary music, it's more like the interpretation of older songs from famous guitarists like Keith Richards. I find amazing because I think he's a really, really one of the best guitar and bass players that are still, I mean, he's a legend, and we don't discuss
Definitely,
that.
definitely.
And there's a version of I think Lou Reed Velvet Underground song. It's called "Waiting for My Man," and for some reason it just gets my attention now. It comes into my memory, which is because the way he interprets it, there's a lot of depth in it, I guess.
So there's a Keith Richards cover song
Yes,
of "Waiting for My Man" from
yes.
really.
Oh, yeah, it is.
Oh, interesting. Okay.
And so again, it's just there are many other ones, but that's
Okay.
that
Yeah,
comes
yeah,
to
yeah.
my mind.
Oh, that's very interesting. We'll check that out. And last, the luxury item for the
I
desert island.
disappoint you because the least luxury interested person I like mountaineering, skiing, maybe. I think one of the really valuable things I own is a race bike. A race bike, and I bought it when Lance Armstrong was such a hero in my eyes. You know, he conquered the cancer, and so with the last money, and I remember my wife was really upset at that time because we were about to buy a kitchen. I was young at that time, and I bought for 3,400 euros, and that's 25 years ago.
Oh my God!
This
Yeah.
Lance Armstrong race bike, and I still use it now. I mean, it's really good quality, and I took good care of it, and there's not too much different to the nowadays bikes that costing 1,500 euros, 15,000 euros.
Yeah.
So that's probably my only really luxury item, I guess.
That's great. That's great. Rainer, thank you very much for explaining all this. We've covered a lot of ground. I think it's much clearer now what this medication, what this drug is, and what it
Andre,
does.
thanks for the invitation, and unfortunately your listener cannot actually see where we're sitting. We're sitting in a room in Slovenia.
In Slovenia. We're sitting in a room where we can see
see
Croatia, the
and
sea,
see-
and the Italian Alps, everything in front of us.
Everything. So next time you have to come to Slovenia in person, I guess.
Thank you. I agree completely. Thank you very much.
Okay, thanks. Bye-bye.